So now we're going to talk about brain protection and support. Obviously, there are many bones that protect the brain. Most of these are the cranial bones, like the frontal, parietal, and occipital bones. There are membranes that protect and support the brain. These are called the meninges, and we'll talk more about those in just a moment. Cerebrospinal fluid also helps to protect and support the brain. It can act as a cushion for the brain, but it does also supply nutrients and water and gases to the brain's tissue. And then there's something called the blood-brain barrier, which helps to protect the brain by helping to selectively transport compounds from your blood supply into your brain's circulation. There are many things that are in the normal blood supply that you don't want in the brain circulation. For example, adrenaline, which is secreted by your adrenal glands that sit on top of your kidneys. You don't want that getting into your brain tissue because that can alter your brain function. Hence why we have the blood-brain barrier. So we'll talk more about each of these except for the bones, which we've learned about in previous studies. So we'll start with the cranial meninges. So to kind of orient yourself to this picture, so here we can see we're looking at sort of a strange kind of coronal section from the skull and brain here. So this would be the parietal bone right here. And the meninges are deep to the bone. So the meninges are found between the bone and the brain tissue. And they're listed in red. There are 3 layers to the cranial meninges: the Dura Mater, the Arachnoid Mater, and the Pia Mater. So here we see the Dura Mater in white, the arachnoid, and then this thin layer that's right on the brain tissue, this is the Pia Mater. The meninges are mostly there to protect the brain from cranial trauma. In other words, if there's some sort of trauma to the head, getting hit by like a baseball or you're in a car accident, the meninges can help to protect the brain from trauma. Though they don't do a whole lot of protection. Your bones are much better for that. So the first layer we're going to look at is the Dura Mater. It actually has 2 layers that are normally pressed together, but sometimes they separate out. Let me take you back to this picture to explain. So you can see here that the Dura Mater is made of 2 layers called the periosteal and meningeal layers. So normally the 2 layers are together. So here we see the 2 layers together, but now the 2 layers have separated out. Right? When the 2 layers separate, a space is formed, and we call this space a dural sinus. And the dural sinuses are going to be filled with blood, used blood. So to come back here, in some places there's a space called a dural sinus that's found between the 2 layers. And the function of the dural sinus is to collect used blood from the brain. In some places, the dural layers fold and invaginate to separate major brain parts. So we can see that here. Here we can see some folds of the Dura Mater that are coming down, and in this case, separating the right and left hemispheres of the cerebrum. Here we can see the superior sagittal sinus. Here's another fold that's helping to form the transverse sinus. Now moving on to the arachnoid mater. Arachnoid means spider. When you look at this white layer, you notice these little cobweb-like structures dangling down. That's where the arachnoid mater gets its name, from these cobweb-like structures. So the arachnoid mater is the middle layer of the meninges, and it does have a space associated with it. These cobweb-like structures are dangling into a space called the subarachnoid space. It's called the subarachnoid space because it's below or deeper to the arachnoid. So the subarachnoid space is technically located between the layer of the arachnoid mater and the Pia Mater. And the subarachnoid space contains cerebrospinal fluid, which we'll talk more about in a bit. The last layer to the meninges is the Pia Mater. It lays right on the brain tissue itself. We can see it right here. It's a rather thin layer, doesn't do a whole lot, though it does help to kind of harness blood vessels in place on the brain surface. Cerebrospinal fluid is composed mostly of water, like most of our body fluids. It does also contain various ions like sodium, potassium, and calcium, and it contains nutrients like glucose and dissolved gases like oxygen and carbon dioxide. The function of cerebrospinal fluid is to both provide sort of like a cushion to the brain, but it does also help to circulate nutrients and gases throughout the brain. We can roughly separate cerebrospinal fluid into 2 locations. inside the brain and outside the brain. The cerebrospinal fluid that's located inside the brain is located in what's known as the ventricular system, named for the fact that we have these very large lake-like structures in our brains called ventricles that contain cerebrospinal fluid. There are about 4 ventricles in the brain, and they're connected to one another through little rivers that are called ducts or ventricular ducts. Please note that we will also find cerebrospinal fluid inside the spinal cord in the central canal of the spinal cord. Talk more about that when we get to the spinal cord later. Cerebrospinal fluid can also be found outside the brain, surrounding all the brain surfaces. And I just mentioned this earlier when we were talking about that subarachnoid space. The subarachnoid space is the space between the arachnoid and pia mater, and it does contain cerebrospinal fluid. The brain very carefully monitors the composition of your cerebrospinal fluid. It knows how much water is in there and how much sodium and how much glucose and how many amino acids. And any changes in cerebrospinal fluid help to alert the brain to changes in the body. So for instance, if your cerebrospinal fluid is suddenly very low on water and high on sodium, that can indicate a state of dehydration. When cerebrospinal fluid is located inside the brain, it's located in that ventricular system. The ventricular system does use ventricles, which are those lake-like structures that contain the cerebrospinal fluid. There are 4 major ventricles of the brain. They're listed as first, second, third, and fourth. The first and second ventricles are also known as the lateral ventricles, and they're located inside each of the cerebral hemispheres. Let me show you this picture. Here we're looking at a side view of the brain, and here is an anterior view of the brain. And this sort of grayish, bluish stuff is supposed to show the ventricular system of the brain. So this curving structure here sort of looks like the horn on a ram's horn. This is a lateral ventricle. Seen from an anterior view right here, this is a lateral ventricle. And you can see how there's a lateral ventricle on both the right and left sides of the cerebrum. Then there's a third ventricle, which is associated with the diencephalon, and a fourth ventricle that's associated with the medulla oblongata. Coming back to this picture, this is the area of the diencephalon, so this would be the third ventricle, and down here is the fourth ventricle. So third ventricle in this picture and fourth ventricle here. When you look at this fourth ventricle, you'll notice that it has 1, 2, 3 openings, and if you count this extension down here, there's a fourth opening. The cerebrospinal fluid that is in the fourth ventricle can either go down the center of the spinal cord, or it can go out any of these openings, which would lead into the subarachnoid spaces of the brain. As we age, our ventricles naturally get larger, but in some disease states, such as Alzheimer's disease, the ventricles can become overly large. We'll talk more about that later when we talk about Alzheimer's disease. And just to show you a different view, this is a horizontal section through the brain, and the arrows are pointing to ventricles in the brain. So depending upon where you make the horizontal section of the brain, the ventricles will all look very different. This is a coronal section through the brain. Here we can see parts of a lateral ventricle here and here. And up here, this is also lateral ventricle. Don't forget these lateral ventricles curl around like a ram's horn. Hence why they kind of have this odd appearance here. This is the third ventricle and the fourth ventricle. Cerebrospinal fluid is produced by structures called choroid plexuses. The sort of red and pink and blue structures in the picture here, those are the choroid plexuses. And the choroid plexuses are made of blood vessels, specifically capillaries, that are coated with a type of neuroglial cell called an ependymal cell. So basically what happens is blood comes to the brain into these capillaries, and then the ependymal cells kind of act as a filter, helping to decide which things are going to leave the blood supply and enter into the cerebrospinal fluid. So the cerebrospinal fluid flows throughout the ventricular system. Eventually, the cerebrospinal fluid will end up in the subarachnoid space, which we see here. And eventually, the cerebrospinal fluid will flow into these bubble-like structures called arachnoid granulations. The arachnoid granulations are structures that bubble into a dural sinus. So this is a dural sinus right here, We find blood in a dural sinus, and we find cerebrospinal fluid in the subarachnoid space. So when the cerebrospinal fluid goes into an arachnoid granulation, the cerebrospinal fluid then leaves and enters into the dural sinus, becoming blood. So this is how we will drain away our used cerebrospinal fluid, is by entering into the dural sinuses. So we obviously have a high requirement for nutrients and oxygen at the brain, and we must have a good blood supply then to go to the brain. The major arteries that deliver blood to the brain are the internal carotid arteries and the vertebral arteries. The internal carotid arteries are branches that come off of the common carotid arteries. The common carotid arteries are the big arteries in your neck. As you can imagine, there are also external carotid arteries. The external carotid arteries tend to deliver blood to like your facial tissue, whereas the internal carotid arteries are delivering blood deep into your head, in other words, to your brain. And the vertebral arteries help to deliver blood to the brain too. If for some reason this blood delivery system is blocked or compromised, then a stroke can occur. A stroke is also known as a cerebral infarction. An infarction occurs when tissue starts to die. And in this case, the tissue is dying because it's become ischemic. Ischemia is when you've had a loss of blood supply to a tissue. And if you lose blood supply to a tissue, the tissue will begin to starve for both nutrients and oxygen. And if it doesn't get those things, it will eventually die. A stroke can be caused by an embolism. An embolism is when you have a clot that formed somewhere else in the body, and the clot broke off and it traveled through the blood vessels and then lodged in and blocked an artery somewhere else. So this could have been a clot that formed, say, I don't know, in an abdominal artery in your abdomen, but then it broke off and traveled all the way to the brain and got stuck right here. As you can imagine, any of the brain tissue that was being fed by this artery is now going to become ischemic and will stroke. Strokes can also be caused by an aneurysm, which we see down here. With an aneurysm, what's happened is the wall of the arteries become weakened and it balloons out, and it's very, very weak, so it's very susceptible to bursting. And should this artery burst, then the blood will leak out, and obviously if the blood's leaking out of the blood vessel, the blood won't be going to where it's supposed to be going in the brain, and that can result in a stroke also. The blood-brain barrier is another protection that we have associated with the brain. This is a way to isolate circulation in the brain tissue from the rest of your general circulation. So there's some selectivity in the transport of compounds out of your blood and into the circulation of your brain. However, there's little or no control over the transport of lipid-soluble molecules. The reason for that is because the structure of the blood-brain barrier is basically just made of cell membranes. So here we see a blood-brain barrier. You have a capillary and it's coated with this strange-looking thing. This is a type of neuroglial cell called an astrocyte. And the astrocyte basically coats the outside of a brain capillary. And to show you another viewpoint, here's a brain capillary, and on the outside here we see the astrocyte foot processes. So anything that wants to leave this capillary and get into the circulation of the brain, it has to get past the astrocyte foot processes first. Well, the astrocyte and its foot process is just a cell, so it's made of a cell membrane. And anything that can simply diffuse across the cell membrane can simply diffuse across the blood-brain barrier. So the blood-brain barrier is no barrier to lipid-soluble molecules. An example is oxygen. Oxygen can easily cross the blood-brain barrier. Another thing that can easily cross the blood-brain barrier is alcohol. Alcohol is also lipid-soluble. So this is why there are those particular effects that we associate with alcohol consumption in the brain. Okay. There are 4 breaks in the blood-brain barrier. These are 4 areas where we really don't have any barrier, and we need these for different reasons. The first 3 all involve hormones. The hypothalamus, the pituitary, and the pineal gland are all areas in the brain that produce hormones. And we need to be able to get these hormones out of the brain tissue and into the blood circulation. And in order to do that, you can't have a large barrier in the way. You can't have this large barrier preventing you from delivering the hormones from the tissue into the blood. So the blood-brain barrier is broken down at those areas. And the last area is the choroid plexuses. These are the areas that produce the cerebrospinal fluid. And again, if we want to, be able to have a lot of communication between the blood and the cerebrospinal fluid, then we don't want to have this large barrier. We do have a different kind of barrier at the choroid plexus, though, and that's due to the coating, of the ependymal cells on the outside of those capillaries.